How to perform a search using the Allen Institute for Brain Science resources. There are 3 search functions in the Allen Brain Atlas resources: a gene search, a differential search, and a correlative search. While many of our resources include these search features, we'll just be talking about the mouse and the human, adult, and developing brain atlases. From our portal page, click the Mouse tab. By default, you can start typing a gene name into the search box and choose from the suggested names. Clicking on the gene name will preview the gene expression. This search is performed in an identical fashion from the developing mouse brain atlas. The results page just looks different and includes a preview of all the stages of development. The Human Brain Atlas has a similar interface and takes you directly to the heat map visualization. For the Developing Human Brain Atlas, or the BrainSpan Atlas, you'll need to first click on the Developmental Transcriptome tab to do this kind of a search. The adult mouse brain gene expression was registered into a 3-dimensional brain space, which allows for unique searching capabilities when you don't already have a gene in mind. You can search for genes that show a similar expression pattern using either the differential or the correlative search features. The differential search is similar in the adult mouse and human brain resources. In the Mouse Brain Atlas, click the Differential Search radio button and select a target structure or structures. By default, the entire brain is selected as the contrast structure. What gets returned is a list of genes that meet the specified criteria. Similarly, in the Human Brain Atlas, this kind of search returns a list of genes with high expression, which is the red data points, in the selected structures. Note, you can search for more specific expression by altering your contrast structures. The BrainSpan Atlas has a differential search not just spatially, but across development as well. This means that in this atlas, you can choose target structures, as well as target stages. Make sure that you review the How to Read a Heat Map video so that you can understand our visualizations with our quantitative data. The Developing Mouse Brain Atlas, because it's not genome-wide, has several different ways to search for enhanced gene expression. You can perform an anatomic or temporal search, but the structures to choose from are limited. Once you find a list of potential genes, you'll notice the rudimentary heat map, with the highest expression in brown and the lowest in yellow over the selected structures and the selected stages. Because of the limited search utility with these structures, the embryonic data was manually annotated, and you can use this annotation to search for fine gene expression patterns. All of the embryonic data were annotated and are searchable by intensity, by density, and by gene expression pattern. This is a Boolean search, so you can add several rows. Once you have found a gene of interest to look at, click on the gene symbol and then note the experimental ID number. This link is available from the PowerPoint slide and will allow you to take advantage of one of the little-known visualization tools that we offer. The last search I'm going to describe is called the correlative search, and is useful when you found an interesting gene and you want to find other genes with a similar expression pattern. Note the gene expression pattern of prodynorphin in the striatum. A correlative search is at the voxel level. And notice how the gradient pattern in the striatum is replicated. The correlative search feature is available in many of the resources in the Allen Brain Atlas resources, including the Mouse Brain Atlas, the Human Brain Atlases, the Developing Mouse Brain Atlas, as well as the Connectivity Atlas.